Hudson. This is
Ben Holmes.
Or is it Ben and Hudson?
So we did 4 experiments. The first one was the reproduction rate at different temperatures. The second one was the vacuole production at different temperatures. And then the third one was how they react in varying ratios of sugar, sucrose, and sugar water. The fourth one was the rate of cilia growth at different temperatures.
For the reproduction rate at different temperatures, we first took 3 samples of the Tetrahymena in solution and put them into test tubes. We then submerged the test tubes into beakers of water so the Tetrahymena could acclimate. One beaker was placed in the incubator in the biolab at 36 degrees Celsius. One was at room temperature and stayed in the drawer back there. And one was in the refrigerator and it was 12 degrees Celsius. So they were all kept in dark areas relatively, except for maybe the incubator lets a tiny bit of light through the screen on the front. But and if someone opens up the fridge, but other than that, they're relatively in dark areas. We came back in a week and we counted the numbers that we saw and compared them with their average number per field that we counted before we changed their when we first put them in the different solutions. And for the results for that experiment, before we changed temperatures, the average number we saw in the samples was for 4 per field. And when we came back to check on them and count, we found that all of the ones at 36 degrees Celsius, so our hottest one they did all, died unfortunately, which was most likely due to the temperature being a little too high for them. Like I said, the highest temperature is 41, but I'm sure 36 is still not ideal for them, obviously. For the average for the room temp was 9 per field, and for the cold one it was 5. Due to the findings, we concluded that our hypothesis was correct in that the colder solution was going to slow down the reproduction rate, slow everything down. But we did not expect for the ones in the highest temp to die, so that is unfortunate that we could not see how they would have reproduced at that temperature. Then for the vacuole production, we used the same process as the one before for acclimating them, except we lowered the highest temperature 3 degrees from 36 to 33, and that seemed to work very well. We took the average time for the vacuole formation, which is 40 seconds, and compared them to our findings. For our findings, we took them out of the test tubes, put them on a slide, and then I added a pinch of yeast to the slides, but I did them one at a time so their temperatures would not change drastically as I'm waiting to do that with nets. And then we would find one of them starting to eat, and then we would count we would time it how long it would take till we could start to see vacuole formation. For the results, our control group at room temperature matched the average time of 40 seconds. The highest temperature group formed vacuoles in 20 seconds, which is half the time of our control group. And then our coldest group took 2 minutes, which also is expected as it's going to slow everything down. So that one took 3 times as long as our room temperature.
Alright, so for the sugar water experiment, basically what we did was we had 3 groups. We had a control, we had one that had 1.25 grams per 50 milliliters of water, and then the third group was 2.5 grams per 50 milliliters of water. And we made the sugar water solution. The control was just straight water with no sugar, and then we mixed them with the water, and we basically wanted to see how they would react to the sugar water compared to no sugar water, and to see if how fast they would die in the different what's the word I'm looking for? Concentration of sugar in those two different solutions. But unfortunately, they all died the next day. So we concluded that sugar and Tetrahymena do not mix very well, and they will die. And yeah, they don't like sugar. They die in sugar, therefore, which is why I think yours died. And my favorite one was the cilia reformation. So on this experiment, what we did was, or what we wanted to do, was knock their little legs off in the centrifuge, and then we wanted to compare how fast they would grow back at 3 different temperatures. So we wanted to do one in the fridge, one at room temperature, and then the other one at that one. Yeah.
At 33 degrees Celsius.
Yeah.
Because that was the one that we found where they could still live and thrive, but not die.
Yeah. So that was the plan. So what we did the first time we did it, well, let me rephrase that. When we started doing it, we threw them all into the centrifuge, and then our plan was to have them in the centrifuge for about 30 seconds, 25% speed. And we did it 2 times, because we had to do that a total of 3 times planned. And the second time, I did not see any difference between them after being spun compared to Tetrahymena I had that were not spun at all. So then I decided to up the percentage of the rate of speed and then the time. We did it again, they still didn't come off, and then we did it up to 6 times, and the last 2 times it was at 100% speed for about 3 minutes the 5th time and 5 minutes the 6th time, and their legs still did not fall off their cilia. And so we concluded that they were too robust to lose their cilia. Therefore, we could not complete the experiment to figure out or see the rate of cilia reformation at the different temperatures.
Thank you. Have a
Thank
good
you.
day.